In this episode, we will talk about how to mimic human metabolism in mice.
For example, neurons, glia.
Let's talk pharmacokinetics for antibody development.
Following the transplantation of hematopoietic cells.
Let's talk how to test if your drug induces a cytokine storm.
Welcome to JAX Tech Talk with the technical information scientists of the Jackson Laboratory.
Welcome, everybody. Thanks for joining us for this JAX Tech Talk, where we're going to be talking about JAXBoy. My name's Kea Ghosh. I'm a technical information scientist here at the Jackson Laboratory. And as my guest, I have Kevin Lomanak, who's a scientific content strategist. So just a little bit about JAXBoy. And let's take a step back and talk about CD45.1 and CD45.2 congenic marker strains. So congenic marker strains are a widely used technique in the immunological toolbox. And let me just show my screen here real quick. And they're used for tracking cells between donors and recipients. The kinds of experiments that they're used for are bone marrow reconstitution, competitive bone marrow transplants, and adoptive transfers. So if we have any non-immunologists in the audience, they might be asking themselves, well, why not just use fluorescence markers like GFP and RFP? There's many of those strains in the repository. And so there's a couple of reasons why immunologists don't tend to favor that approach. One is that you have a little more flexibility using this congenic marker approach because you can basically use any fluorophore. There's many different commercially available antibodies to these congenic markers, CD45.1, CD45.2. And so if you have those common, red and green channels earmarked for some of your other markers that you're using, you're trying to track in your immunophenotyping experiments, for example, rare populations, you don't have to redesign your antibody panel then. So it just gives you more flexibility. Another kind of maybe more profound reason is that anytime your cells are expressing a foreign protein, that is energetically expensive, and you can have off-target effects, including the immunogenicity of the foreign protein itself. So GFP is immunogenic, right? It has to be, otherwise how would we have anti-GFP antibodies out there on the market? So the disease areas and therapeutics that these techniques are used for are, well, this has particular relevance to hematological disorders such as anemias, malignancies, but really to any disease with an immunological component, even if the etiology is not immunological per se. So we've had these congenic marker strains such as Pep-Boy and others from other vendors out there on the market for a while. Why are we introducing a new one, i.e. JAXBoy, right now?
Yeah, so I think that's a really good question. And the more I've worked on this JAXBoy project and thought about that question, the more excited I am about the sort of concept behind it. I think, so in short, the answer is because we can, and I think because we should. So what do I mean by that? So the older strain that we used to use, what we've traditionally used was Pep-Boy, and I think you've given a good summary there of kind of the uses and what it's for. So I want to take a step back first and talk about how we made that. So we needed a tool that we could use to track cells that were transplanted from one animal to another. And we picked this marker that we thought wouldn't have so much impact, but we could attach things to it, antibodies and such. And as you said, you can attach a fluorophore to that. We can track those cells. To make that thing, the scientific field used the tool set that they had at the time. And that was essentially making one of these congenic markers. They took one mouse and they said, SJL expresses this CD45.1 marker and Black 6 expresses this CD45.2 marker. And what I want is to take this CD45.1 marker and put it on this mouse that otherwise would express CD45.2 and have everything else the same as that Black 6J mouse. So they did that through successive backcrossing because that was the way that we knew how to do things at that time. And I think everybody's reasonably familiar with that concept of congenic mice, that you take your gene or region or marker of interest, whatever it is, and you backcross it. And as you do those backcrosses, you're typically genotyping based on just the presence or absence of that marker. What we didn't have the tool set to do at the time, some of these tools existed and they were just cost prohibitive, but we've gotten better and better access to them, is we didn't think enough about the region around that. So when you do that back crossing, the whole idea is generation after generation, the rest of the DNA throughout that animal gets less and less. And you move from that SJL where it was 100% in the beginning down to less and less and less. And it, 10 or 20 generations, you're talking 99.999% black 6J DNA is the idea, and you just have your little CD45.1 marker. Well, the reality is that it doesn't work that way. You don't get perfect recombination events. You don't get recombination randomly through those back crosses exactly at the start and finish of that CD45.1 region, which is really just a single nucleotide that distinguishes between the 0.1 and 0.2, A or B. So that tool has been really, it worked though for a really long time. The Pet Boy mouse and other versions of this, I know there are other commercially available versions where people have done a similar thing. They all kind of approached it the same way using this back crossing. They've been good enough, but we're at a time in science where we have a different technological toolbox. We have different abilities now where we can make something better. Instead of a congenic thing, we can make a co-isogenic thing. And if you think about the purpose of this model and what it was always supposed to be, that's what it was supposed to be. That's what we always wanted, was this co-isogenic version where everything else about it is 100% C57 black 6J except this one marker. We have that tool set, and I think JAX has a really great model generation core that we use internally. They're available externally as well. And I think it's our responsibility to leverage that tool set to the advantage of the scientific community. And so we identified a thing here, which was a good opportunity to leverage that tool set and make something better for the community to use. So what do I mean by better? There was a paper recently, I think 2-3 years ago, from the lab of Amy Weinman at the University of Alabama. That paper is Chisholm. Oh, good, you got it. It's Chisholm et al. It was an immunity, where they looked at, I think, the 3 most common commercially available C57.1 black 6 mouse models, or LY 5.1. The nomenclature on that marker has changed a little bit over time, but they compared them from several vendors, and they did this type of genome scanning where they got in and really dug into not just that CD45 marker, but the genomic region around it, and looked at what's there. And what they found was, not only is there a pretty substantial artifactual chunk of SJL DNA surrounding it, but it's different from model to model. So if you're using these mice and you're thinking, well, this week I used Pet Boy from JAX, and next week I'm going to get a different one from a different vendor because they have more availability or something like that, you should also keep in mind, I think this is a really critical paper. I think they identified a really substantial issue here that I hope people appreciate, which is they're not the same. What they identified was several genes in the surrounding region around that CD45 locus that had carried over from SJL, and they were able to identify that they carried over from SJL because they differ from the version of that gene that is expressed, the isoform that is expressed in C57 black 6J. So if you're looking here at this sort of schematic of what they found, what I think the immunological community will quickly recognize is there's some really important genes in that region. There's PD1, there's CXCR4, there's IL-10. So there's some genes that we know in and of themselves have an impact in how you might use these in work with immunology. But the other thing that I think is underappreciated is there's probably some modifier genes and some regions in there that impact not only that gene itself, but other genes in the surrounding area. So I think what they did for us was really strongly identify, yeah, there's some genes here that aren't resolved to black 6J the way that we would want them to be for this tool to be a truly perfect tool and be the co-isogenic thing that we wanted it to be.
Thanks, Kevin. And I also want to tack onto that and just note that CD45 itself has a great deal of biological relevance. So it's expressed by pretty much all hematopoietic cells, except for red blood cells, plasma cells. And it's a member of the protein tyrosine phosphatase family of transmembrane signaling proteins. It's thought to modulate signaling signals emanating from integrin and cytokine receptors. And just more broadly speaking, the balance between kinases and phosphatases, not just CD45, but those broad classes of transmembrane proteins, the activity can, or alterations in the activity rather, can result in imbalances leading to immunodeficiency, cancer, autoimmunity. So the regulation of CD45 itself, in addition to all of these genes that are in this pretty long SJL-congenic interval, I believe they suggested it's about 40 megabases, which is pretty big considering how much congenic backcrossing has gone on. That region presumably contains the regulatory regions that affect the CD45 expression itself. And so probably a relatable example of the relevance of CD45 regulatory expression is CD45 low and CD45 high cells. That's a frequently used gating strategy for immunologists. For example, CD45 high cells are often used as an inflammatory gating strategy. So it's quite relevant what's going on with all these genes that they've identified in this congenic region, but also the CD45 molecule itself. And there's a couple of really nice papers that we're highlighting here in case you'd like to read more about this. We don't have time to get into all the details, but this Mercier et al. Paper and Waterstrat et al. Paper, these are both really nice reviews that talked about some of the empirical evidence out there that the congenic strains are somewhat of an imperfect system for tracking donor and recipient cells. And that's due to things like defects in homing, engraftment, and persistence of the subsets when you're transplanting cells from for example, a traditional strain like Pep Boy into your recipients. And these are all things that are really very much at the heart of what you're trying to measure, right? Persistence, engraftment, those are all the things that you're trying to look at in a competitive bone marrow transplant experiment, for example. So I would really encourage you to take a look at this paper. The mechanisms are multifaceted, or the possible mechanisms are multifaceted that they review. But probably the most relatable mechanism that I'll just sort of touch on here is there is some immunogenicity of the isoforms itself to the non-native recipient. So there could be some rejection going on, and that's something that's seen empirically, but that might be one of the mechanisms, is that there's the immunogenicity of the different isoforms. So one thing I'd like to ask is, now that we have JAXBoy, is there any internal JAX data that would speak to JAXBoy's utility in these types of experiments?
Yeah, of course. So I'm glad you mentioned the Mercier paper there, because I think that was a really good proof of principle for us going into this, and it made the case for creating this model really easy, because they did a similar thing, right? They used CRISPR-Cas9 to generate a co-isogenic CD45.1 Black 6 mouse, and they looked at some of the downstream effects. That model, unfortunately, was never made commercially available, as far as I know, so it's hard to get, if at all. So yeah, we went and recreated something. We made the thing the way that we wanted it to be, and then of course the first question is, were we successful? Did it work? So internally, a bunch of people have worked on this. Maximiliano Pressa, I think, did a bulk of this work. So we looked at, I think we started at the point of what is one of the most likely uses for this mouse model. What do we think people want this thing for? Why would someone buy this mouse and use it? And one of the most obvious cases is competitive bone marrow transplant. So the idea here is you breed two mice together, and you make a Black 6 mouse that is double positive for CD45.1 and CD45.2. You irradiate that mouse, you ablate the cells that are in the bone marrow, the native cells, and then you're engrafting in equal parts of progenitor cells, hematopoietic stem cells from a regular, a wild type C57 Black 6J mouse, and from your what should be a Black 6J mouse that expresses CD45.1. So you have some cells that are coming expressing CD45.1, some that express CD45.2. And if your CD45.1, if your Pep Boy or your JAXBoy or whatever model it is what it's supposed to be, that mouse that you're engrafting those cells into shouldn't be able to tell the difference, because everything else should just be C57BL6J. So it shouldn't make a difference, and you're putting in equal parts of cells. So as those cells that you've engrafted home to the bone marrow, and they engraft and they grow up and expand and they move out, what you should see is equal parts CD45.1 and CD45.2 cells. When we looked at the PetBoy mouse, our traditional congenic model, what you can see is pretty quickly, and so there's data here looking across 16 weeks, we track this engraftment over time, it's not equal. That mouse that host that you've put it into can tell the difference between those cells, and the cells that came from a BLAC6J mouse that expresses CD45.2 are thriving and engrafting better, not only initially, but certainly over time as well, than the CD45.1 expressing cells that you got from a PetBoy mouse. Now, if we do the exact same thing using JAXBoy, where it's co-isogenic now instead of congenic, we've gotten rid of all that extra SJL. So really and truly, this thing should be 100% almost C57BL6J, except for that one nucleotide that determines CD45.1 or 0.2. And so we looked at the same thing, and there's two things. Strikingly, right off the bat, there's better engraftment. There's no significant difference right at the 4-week time point between CD45.1 and CD45.2 expressing cells. Over time, it's also a much more stable engraftment. You can see with PetBoy, they sort of divide over time as some of those cells thrive more than others. It's a really, really stable engraftment that we see with the JAXBoy mouse. This, I think, is a really big factor. So you're getting a better engraftment. The cells that you wanted to put in that mouse are thriving in that host. And so what that means, and I think this is really going back to the question of why did we make this, I think this is an important factor for a lot of reasons. But one of them is it's a 3 R's compliancy issue. If you have better engraftments, if you know I need X number of cells and I'm going to do these engraftments and I'm not going to get 100% engraftment because the cells aren't going to take, you're going to have to use more mice, or you're going to have to use more cells. This is a more efficient way of approaching this because you get those better engraftments. You get the cells that you need. You can do fewer mice in your studies. You can take a tighter approach to designing your experiments. So I think there's other data, and certainly we'll get some of this on the data sheet so people can have it there and show it to their lab and talk about it in the future. But I think this really illustrates the point, which is that bit of SJL DNA that remains in a PetBoy mouse or in any other mouse that was made through this traditional back crossing to be congenic. It does have an impact. It's not the same as it being a B6 mouse that just expresses CD45.1. Okay, so what I came here also with a purpose, and that is, as I said, there's a lot of uses that we know people take these mice and they put them into use in their lab. In terms of planning how to meet those needs, I would certainly like to take this opportunity to ask a couple of questions about how people might also be using them. So the first question, I'm going to throw this up there, is, are you using CD45.1, CD45.2 double positive mice in your research? So essentially doing something like this competitive bone marrow transplant. Are you taking these mice and immediately crossing them to a BLAC6? That's something, in theory, if there's a lot of demand for that, we can maybe make that a little bit easier for people to get those mice. So we would certainly want to gauge the enthusiasm for such a thing. So I'll give you just a couple of minutes there. It looks like there's a fair number of people who are doing that. Not everybody. People have found a whole lot of other uses. All right. And a couple more seconds. All right. Thank you, everybody, for that. And then I have just one more question here that I wanted to ask you. So that is, when you get these mice, for folks who are out there using PetBoy or hopefully JAXBoy in the future, do you ever cross these to something other than just a BLAC6 wild type, or just use it as is? Are people crossing these to other potentially immunological strains such as an OT1 or OT2, TCR transgenic, CD90.1? And part of the reason we want to know about this is, I think this is again, another issue where having JAXBoy be a co-isogenic and getting rid of that additional SJL is really going to come out later on in science, because if you're crossing these to other things, and let's say the other strain that you're crossing it to was also some kind of congenic, or maybe it was a targeted mutation that wasn't made using CRISPR, that was using traditional transgenics, where it was done likely in a 129 ES cell, and then it was back crossed to be congenic. Well, now if you're using something like a pet boy that has that residual SJL, now when you're talking about these modifier genes, other genes that are within that residual artifactual DNA region that we talked about a little bit, now you're compounding those effects. So now you have a black 6 congenic mouse, but it potentially has some SJL to it. It also has some 129 to it. So I think that's where the value of JAXBoy is really going to shine in the future, because you get rid of some of those artifacts that can impact your data, and the analysis and the interpretation of that data in the future. So thank you everybody for that information. That's going to be really helpful for us.
Yeah, I'm glad you asked that, Kevin, because I feel like in my role here as a technical information scientist, I do get that question once in a while on the phone, both of those questions actually. So it'll be good to get a survey of how important this is to our viewers. So let's say a lab is convinced that JAXBoy is the scientifically superior approach. What kind of assurances, if any, can JAX offer that the transition will be smooth, logistically speaking?
Yeah, so excellent question. I think that's the big lift here is, because to me, scientifically, it makes all of the sense to switch to a JAXBoy. But there's certainly the question of, and this is one of the caveats of how scientific research works, is it's always built on the foundations of what's been proven, what's been tried before, what do we know. I can tell you we have some data that says right at the out, I mean, there's the data that we're showing here. We've also looked at, just common logistical questions of breeding efficiency. Things like, can you use the same reagents, the same husbandry? It's the same background strain, of course, but perhaps a little bit more of that background strain. I think one of the big questions we dug into was whether or not you can use those same commercially available reagents, because that's always a question. I've already bought my antibodies. They're sitting in the fridge. So we looked at the most commonly used reagents and found, yeah, they work perfectly well with JAXBoy. So if you already had invested in those antibodies, those will continue to work for you. And I think we have some data. Kaya, do you have a slide with body weights? Yeah, if you look at these two and compare them side by side, the way they grow, the way they present, I mean, and they should just present as essentially a black 6J mouse. They're really consistent. There's some other data there that, again, we'll put up on the strain data sheet for people to look over. The other thing that I want to point out is, and this data set was a little bit too complex for this particular view, but we looked at the expression of different immune cell populations and compared them between C57 black 6J, the congenic pet boy, and the co-isogenic JAXBoy. And that was one of the data sets that really struck me, because what we found is JAXBoy looks a whole lot more like C57 black 6J than the pet boy does.
And this is just in naive mice, is that right, Kevin?
This is just in naive mice. These are just the cells expressed in a wild type mouse if you just pluck them out of the cage and take a look at what's in their blood.
Looking forward to seeing that data. That sounds really good.
I actually just saw that data last week and I was really excited about it. So we're going to polish that up and get that on the website as quickly as possible so that other people can take a look at that, because I know that those values are really important to people.
Yeah, absolutely. And I just want to add, because you had mentioned antibodies, there's a variety of different antibodies on the market for CD45.1, CD45.2, at least the CD45.1 commercially available antibodies are by and large, if not exclusively, produced from the A20 hybridoma clone. So whatever antibody you have, even if it's not the exact one that we used in our tests, is expected to work for that reason. So just another It's just another thing to kind of allay your trepidation, because I understand anytime you're thinking of switching something up in your system, of course, as scientists and lab scientists, we're always thinking, what can go wrong? You know, is this going to be an easy transition or not? And we really believe it will be for you.
Yeah, I think it's an exciting thing. I think with this one, it's been easy to make the case for why it should be made, how it should be made, and it's been pretty easy to test it and see, is it effective? Does it do what we hope it will do? I think we've tried to generate the data that we think will make it easy for people to transition to what we genuinely believe is a better version of a tool that we weren't able to make before and we are now. For me, I think one of the next big questions looking forward is, what other types of models should we apply this idea to, of taking an older technology that may have been a little bit limited in its utilization, and making better versions of them using more modern science. So I'm excited to see where this leads in the future.
Sounds great. Me too. Well, thank you so much for your comments, Kevin. That was really interesting. And I want to thank all our viewers for coming today. We're keeping an eye on the questions, and I see that there's a number that we didn't have time to get to today. But if that's the case, we'll certainly follow up with you afterwards. And thank you so much for coming. And if you have any questions about JAXBoy or congenic strains in general, you can always write to us at micetech@jax.org. That's the address of the technical information scientists. So once again, thank you so much, and I hope you have a great rest of your morning or afternoon.
Thanks so much for having me, Kaya. Thanks, everybody.
Pleasure.